Okay, thank you, Firuz, for the great introduction. Good afternoon, everyone. I'm Shahab Rahimi, part of the product development team in Ardena. We specialize in manufacturing, development, and manufacturing of oral solid drug products. Our site is actually located in Somerset, around 20 to 30 minutes drive from here. It used to be Catalan's Center of Excellence for hot melt extrusion. We support our clients from early phase product development all the way to clinical manufacturing, clinical and commercial manufacturing. So today I'll be talking about solubility enhancing technologies for poorly soluble drugs, mainly focusing on spray drying and hot melt extrusion and the approaches we take from early stage development and formulation screening all the way to transfer and scale up to manufacturing. So the current estimate is 70 to 90% of new chemical entities, NCs, coming out of discovery pipelines suffer from poor aqueous solubility. 40% of the marketed products belong to BCS class 2 or class 4. Expanding on the BCS classification, we have the developability classification system, or DCS, which is basically a modified BCS categorization. You have the dose solubility on the x-axis and then permeability on the y. The DCS class 2 is divided into 2A and 2B. Compounds in class 2A, they are dissolution rate limited. So typically, technologies that are applicable for 2A compounds are those that are based on increasing the surface area of the drug substance, such as nanosuspensions. Typically, you have API, polymer, and surfactants. There are different manufacturing technologies, top-down, such as media milling, basically to reduce the particle size, or bottom-up, such as solvent, anti-solvent systems. And then another approach is micronization, for example, jet milling to reduce the particle size of API. And that API can either be directly filled into capsules or blended and then filled into capsules. A good dosage form for preclinical or FIH, first-in-human studies. And then we have DCS class 2B. These are solubility limited compounds, so reducing the particle size, increasing the surface area may not be sufficient to improve the solubility and absorption. And then DCS class 4 compounds which suffer from low permeability as well. Technologies applicable to these compounds are amorphous solid dispersions, mainly spray drying and hot melt extrusion, which I will discuss in more detail, and then lipid formulations. We had a nice presentation earlier on how to formulate these SEDs and SMEDs, and actually some advanced formulation technologies can combine solid dispersions with lipids to address both the solubility and permeability challenges. Just quickly going through the concept of solid dispersions, it's basically relying on the amorphous form of API, which is in higher energy state, and there is a less energy barrier for solvation. Typically, these amorphous forms of API are stabilized within a polymer matrix by solvent evaporation methods like a spray drying or melt fusion-based methods such as hot melt extrusion. The graph on the right shows the solubility of crystalline drug. And then for this amorphous form, you can either have a sharp increase or a spike in the concentration followed by precipitation of the crystalline form, or ideally, with the proper formulation and choice of components in the formulation, you can extend that extent of supersaturation to provide a wider window for absorption. Now, from 2012 to 2023, a recent report showed that there were 48 approved amorphous dispersion products. 61% of those violated at least one of the Lipinski rule of 5 aspects, so molecular weight, log P, or the number of hydrogen bonds. And if you look at the graph, there is a great portion of compounds with high molecular weights, which basically means they are mostly falling into DCS class 4 with low permeability, and high log P, which is basically being highly lipophilic molecules. Makes these very challenging in terms of designing the formulation. Now, with these NCEs and development of the oral solid product, there are some challenges, especially with early phase formulation and process development. Typically, the timelines are very aggressive. The compound needs to get to clinic as fast as possible. The API supply is usually very limited. In a lot of cases, we work with milligrams of API, and we have to have methods and procedures to be able to get meaningful data by using those small quantities of API. And then I'll also show some of the strategies that we use for once we develop a formulation, how we scale it up within the framework of QBD principles for GMP manufacturing. Now, looking at the spray drying process, not going to go into too much detail, but basically you have a solution of API with a polymer or different ingredients such as solubilizers, surfactants. It's sprayed with an incoming stream of hot gas, typically nitrogen, into the chamber. It's atomized, droplets dry, and then collected in a collection vessel. Now, spray dryer can run in either open loop mode or closed loop, where the gas basically recirculates in the system, and it goes through a condenser to remove the solvent. At different stages of development, there are different types of equipment. For example, for early stage formulation development or screening, a Buki is a common one, capable of handling a few grams to maybe tens of grams. And then phase 1, first in human studies, PROCEPT or MS-35, and then going to larger scale. Typically, with the larger scale spray dryers, they mostly run in closed loop. So the thermodynamic conditions of the process are different with the open loop spray dryers. And you can see how the scale varies, going from hundreds of grams for equipment for phase 1 FIH to hundreds of kilos, for example, in PSD4, which is a commercial scale spray dryer. Now, looking at the pre-formulation and formulation screening, typically the first step in the approach that we take is to really understand the physical attributes, chemical and physical attributes of the API. There are different techniques, X-ray, DSC to understand the thermal properties of API, log P, melting point, glass transitions. All these attributes are essential to be able to design a formulation that can improve the absorption in the preclinical studies. Then, we can utilize in silico tools. Basically, there are a number of thermodynamic models that we can utilize to assess the miscibility of the API with respect to different AST polymers. An example here is the Hansen solubility parameter. Using the chemical structure of API, we can estimate the Hansen parameter and use, in this case, Flory-Huggins equation to basically, without using any API, construct a theoretical phase diagram. You can see for different polymers at different drug loads. And typically, this really guides the formulation instead of randomly picking polymers and screening a large number of formulations. just in the beginning, without using any API, we can get a sense of how these different polymers with different chemistries, cellulosics, PVP-based polymers, can interact with that particular API structure. And also, at what drug load you see the minimum Gibbs free energy of mixing, which is indicative of maximum thermodynamic miscibility. Then we have screening experiments. For example, for spray drying, one of the most critical aspects is the solubility of drug substances in organic solvents. Typical solvents used in spray drying with low boiling point or mixtures of solvents. And then smaller scale screening experiments, for example, microtubes, microevaporation techniques, utilizing less than 50 milligrams of API, and then get a quick reading with tools such as micropellate readers or basically fiber optic. Two important types of experiments we run are SSKD, or supersaturated kinetic dissolution. Basically, that shows us how these different combination of API with polymers can improve the kinetic solubility of API. And then we can also look at liquid-liquid phase separation experiments, where we introduce a portion of a stock solution of amorphous API into a solution of different polymers to see how it can prevent the crystallization, or basically prevent the API from crashing out of solution. Now, it's again, as I mentioned, it's critical to understand what challenges we are trying to address for that particular drug substance. One important graph is the one that I'm showing here. Basically, you have the ratio of melting point to glass transition temperature on the y-axis, and then you have the log P on the x-axis. So as you go up, basically with increasing Tm to Tg ratio, that indicates that the API is a poor glass former. So if you convert it to amorphous form, that amorphous form is not very stable. What may happen is in the top right graph, you see even if you can basically get a spike in the concentration, most likely that parachute effect would be very weak, and you would see an immediate basically decrease in the solubility of API, basically crashing out or recrystallizing, which again really shows us what formulation strategies we need to utilize to basically obtain that sustained supersaturation or extend that parachute to improve the absorption in vivo. Another case is on the lower right side of the graph, which is basically for the high log P molecules. These are highly lipophilic, especially with increasing drug loads. We may end up having patches of lipophilic or drug-rich phases on the particles of solid dispersion, whether HME or STD. And what may happen is you would see that the dissolution rate will actually go down, which again suggests that we have to come up with strategies to overcome this and improve the dissolution rate in those formulations. Now an example, looking at the approach. So initially it started with the screening experiment. We see that in this case there are 3 different polymers, HPMCAS, PVP, VA64, and Soluplus, providing the highest supersaturation. Again, this was only using milligram amounts of API to basically do this formulation screening. We looked at different drug loads, took 20% and 50% drug loads to the next stage, which was proof of concept. We spray dried a prototype on Buki. And I have some data for T0 and up to 2-month stability at 40C, which shows that the formulation remains amorphous. Now, So after this early phase proof of concept work, when we go to the stage of developing an actual process in the context of QBD, we really need to develop a good understanding of how material attributes and critical process parameters will impact your CQAs or your product's critical attributes. In terms of spray drying, some of the material attributes are listed here: solubility of API with the polymer, the miscibility, viscosity of the feed solution, and also the solid content, which is again, it's related to the solubility of the API. CPPs, you have independent process parameters, such as feed rate or a spray rate, inlet temperature, atomization process, whether it's the nozzle size or pressure. And then you also have dependent process parameters. And these are scale independent, which means that as you go from lab scale to production scale, equipment is essential to be able to maintain these scale independent parameters. For spray drying, the critical ones are usually droplet size, the outlet temperature, and the relative saturation of the gas at the outlet. And then critical attributes for the spray dried material is usually if there is any residual crystallinity, the glass transition, residual solvent, which impacts the physical stability of the product and also dictates the downstream secondary drying, which again, it can impact the stability of the product. And then you have assay, particle size, and process yield. Now, I have an example of how a typical process space can be developed for a spray drying process. Basically, you have feed rate versus inlet temperature. And then you have these isotherms of outlet temperature. As you can see, with decreasing the feed rate or increasing the inlet temperature, the outlet increases. Also, I have the dashed lines on the residual solvent in the system or relative saturation. And you can see the area that's shown by the red lines. Basically, your inlet temperature boundary would be decided based on the evaporation capacity of the spray dryer. Usually, as you go up in the scale, there is a minimum feed rate that we want to maintain to have an efficient process. And in order to achieve a certain outlet, we can set the inlet temperature. And that's line number one. Line number two is, again, dictated by the scale, basically the feed rate that we are spraying. And then you have the boundaries number 3 and 4, which are dictated by the outlet temperature and maximum relative saturation at the outlet. So once we identify these variables, we can design a control strategy to maintain the process within that constraint of this boundary to ensure we get the desired quality attribute consistently during manufacturing. Now, changing gears a little bit, going to hot melt extrusion. Again, technology to produce amorphous dispersions. Basically, you have the API and polymer going into the twin screw extruder. Depending on the melting point of API and process temperature, we can run this in either solubility or miscibility regimes. Typically, there is a misconception that the API needs to melt during extrusion, which is not the case. We have a lot of cases where we can successfully develop products much below the drug melting point. And basically, so extrusion and then pelletizing to get the solid dispersion pellets, milling into powder, and then downstream work to develop tablets or capsules or granules to be filled into sachets. Again, some pre-formulation techniques, usually for HME, we have DSC and TGA to evaluate the thermal properties of the drug, and also hot stage microscopy to actually be able to visually see what's happening to the drug as we heat it up in presence of polymers or polymer mixtures with surfactants. I have listed here a few challenging cases for developing a product using HME. First one is a high melting point. So this particular drug had a melting point of 300 degrees. You may know that there is no pharmaceutical excipient that can be processed at that temperature, but we have been able to process this API at much lower temperatures and convert it to amorphous form. We have APIs that basically degrade upon melting. The second graph shows an overlay of TGA on the DSC, and you can see as soon as API melts, you see a huge drop in the TGA graph, indicating that the material is decomposing. And then the third scenario is APIs with very high tendency to recrystallize. So a typical heat-cool-heat cycle shows that during the cooling, there is a huge exotherm, indicating that the API recrystallizes completely back to its original crystalline form, even at high cooling rates. Again, these are the challenges that you may see because, let's say with the third case, even if you can convert the API to amorphous form during extrusion, if the formulation is not right, the API can immediately go back to its crystalline form after the process. Some of the techniques and approaches that we take for formulation screening with HME, we use DSC basically to monitor either the solubility or miscibility of drug with the polymer, typical heat-cool-heat cycles, to identify if we can see a melting point depression, and basically we can dissolve the API into the polymer below its melting point. And hot stage can be utilized along with DSC to actually visually see. In this example, you can see the black specks pretty much disappear around 180, which is indicating that the API is dissolving in the polymer. And then we have melt rheology studies, basically evaluation of viscoelastic properties of the polymer API blends to identify to basically understand the extrudability of the blend and how the material behaves as a function of shear rate or temperature in the extruder. In terms of using material sparing approaches, we utilize VCM, vacuum compression molding, which is a technology to mimic hot melt extrusion in a very small scale. Typically, you can use only 100 milligrams of API polymer mixture. Basically, it compresses the material at high temperature under vacuum into discs, VCM discs. These discs can then be used either milled into powder or studied directly for X-ray dissolution, and we can also supply this powder for animal studies. So, it's very important to be able to, develop a micro prototype for preclinical animal studies using only milligram amounts of API. After the formulation screening, we typically construct a phase diagram. Basically, this is a temperature composition phase diagram. The upper boundary is the degradation of API, where your process temperature is limited. Your lower boundary is the glass transition temperature, so you need to process above that. And then you have that solid black line or curve, which is the solubility obtained from hot stage or DSC. And in order to show that how well it correlates with the actual process, this is an example where we basically looked at an API with a PVPVA64 polymer, and we took 4 points on this phase diagram. 2 points on the solubility line, number 3 in the below the solubility line, and then 1 in the green area, which is your HME process space. And on the right, you can see number 1, 4, and 3, basically the pink, green, and brown on the top. They show residual crystallinity, whereas the prototype made at number 2, which is in the green zone, shows the amorphous conversion of API. This is another example. Again, developing a formulation for a high melting point API. The melting point was around 250. We looked at different combinations with DSC and hot stage, and you can see on the hot stage in the PVPVA 64, actually we looked at different compositions. Here I'm showing the 30% drug load formulation, and you can see the black specks disappear around 200, a little bit less than 200. Again, melting point being 250, we are able to dissolve the API more than 50 degrees below the melting point. So we took this formulation on a Pharma 11. It's a benchtop extruder, twin screw extruder with a batch size of 30 to 40 grams. And we actually processed a formulation with the PVP and SoluPlus, and you can see the process temperature profile is 70 to 60 degrees below the melting point of API. And both formulations came out amorphous. The lead formulation on the SSKD, or kinetic solubility, is the green one, which is the PVPVA, showing a significant improvement and sustained supersaturation for this formulation. Now, when it comes to process development and scale-up, we can support projects from preclinical development using benchtop extruders all the way to 18 and 27 millimeter extruders for commercial manufacturing. Typical throughputs, few hundred grams on the bench scale, up to 5 to 15 kilo per hour feed rate on the 27 millimeter extruder. And then again, same concept, evaluating the critical process parameters and critical material attributes and how they impact your quality attributes of the product. For the extrusion process, the 3 scale-independent parameters are melting point, residence time, and a specific mechanical energy. Each one can be studied separately in its own design space. For example, having the throughput or feed rate with versus a screw speed. You see that for the melt temperature, as you increase the screw speed or reduce the throughput, the melt temperature increases. Residence time is the other way. So with basically reducing the screw speed or reducing the throughput, the residence time, or the time that the compound retains in the extruder, increases. So by doing these experiments, running samples, understanding where the API degrades, where the API shows residual crystallinity, we can actually design the process space with the boundaries of melt temperature and residence time, and then look at the specific mechanical energy, which is basically a measure of how much energy is being input to the material. And you can see that green area shaded with green color is where you want to basically control your process to be basically to constrain it within your boundaries of melt temperature, residence time, and specific mechanical energy. Now, just to summarize, hopefully I was able to show you some of our material sparing and high throughput screening approaches for early phase formulation development for spray drying and HME. I showed how it's critical to have a good understanding of the physicochemical attributes of the API in order to be able to come up with a formulation that's safe, effective, and scalable. and then how these CPPs, critical process parameters and material attributes can impact the quality attributes of the product, and how it should be studied in terms of a scale-up and transfer to production. With that, I'm going to pause here. Thank you for listening, and I'd be happy to take any questions.
Questions?
Hi. Yeah, nice talk. The question I had is the QBD for scale-up in HME you've studied is just drug and excipient. Do you think when you add excipients that can lower, like plasticizers and maybe drugs that act as plasticizers, would that have more of an influence on lowering the temperature, or is it just screw speed, feed rate, and all those? So how much of an effect do you think it's because of just the composition due to polymer and drug, and how much would those additives play a role?
The impact is significant. A lot of times you have processing aids, surfactants or solubilizers to basically get two functions. One is to lower your process temperature, and especially if you are processing in the solubility regime where you have to dissolve the API in the polymer, those surfactants or solubilizers can actually accelerate that. And it may enable you to process and dissolve the API at much lower temperatures. Most simple formulation would be a binary mixture of API with polymer, but a lot of times there are different components that needs to be added to either alter the process conditions, or some examples I showed with if API has a high recrystallization tendency or highly lipophilic molecules. A lot of times, we combine with lipidic excipients to later on address those challenges during the solution of the AST.
Thank you.
Sure.
Thanks, any additional questions for Shahab?
Basically, what they were doing with multi-particulates earlier, adding lipids to do sets and spins, you can do the same with probably not extrusion. You just add them inside as a plasticizer that helps you process, but at the same time, it ends up to help with bioavailability at the end. The Calitra case, as a matter of fact, if you look at it, it was a soft gel lipid-based formulation. They changed it in a tablet. But if you look at the inactive ingredients, they kept the lipid inside that tablet.